Namaskar. I'm Harpreet Kaur and I welcome you all to our live and interactive session. You are watching this special session of ours, yes, our webinar series titled Listening to Learn, यानी कि सुने और सीखें. Today we are in the 95th edition of this webinar series, and this series is brought to you by Department of Education in Science and Mathematics, NCERT, New Delhi. Well, this series is about all the different topics that we take and eminent personalities who share their views on the same. Well, we have talked about health, we have talked about sea, and we have talked about chemical compounds as well and whatnot. I mean, this is the series which can give you the answer for most of your questions. So what are you waiting for? Today we have got you yet another interesting topic, and that is Khorana and the genetic code. Well, it is quite interesting. So who was this Mr. Khorana, an eminent personality, and what about this genetic code? Why we are going to talk about it today? Well, we will reveal all the details in a while, but before that, let me tell you, you are watching this session of ours on PME with their channels 9, 10, 11 and 12. Yes, the session is live and interactive, and we are streaming live on our YouTube handle NCERT Official also. You can also participate in the session with your questions, suggestions, feedback. If you want, any particular topic to be discussed under the series, do let us know. And take down our phone number now, which is 8800440559. We also have 4 email IDs for you, but I'm mentioning here only one, that is dth.class12@cit.nic.in. And you can always go to the live chat box of our YouTube handle NCERT Official for your questions, suggestions, and of course your feedback. Well, since we are in the 95th edition today, that means we have conducted a lot of editions earlier also. So if you want to watch those sessions, you can definitely log on to this particular link, which is ncert.nic.in/webinar_series.php. Now we are joined by 2 eminent personalities. Yes, so one is our guest and another one is the man behind this entire initiative. I will introduce him to all of you and vice versa, and he will take the conversation forward. So let's welcome Dr. Gagan Gupta from DESM NCERT. Sir, namaskar and welcome to the session.
नमस्कार हरप्रीत। दोस्तों, दिल्ली का मौसम सुहाना हो चला है। बाहर थोड़ी थोड़ी धूप खिली है, 14 डिग्री सेंटीग्रेड तापमान है, और आज जाड़े के बाद, जाड़े के दरमियान इतनी अच्छी धूप मिलना दिल्ली वालों के लिए सौभाग्य की बात है। आज हमारे गेस्ट प्रोफेसर उमेश वार्ष्णेय बेंगलोर से जुड़े हुए हैं। मुझे पूरी उम्मीद है कि बेंगलोर का मौसम भी इतना ही सुहाना होगा और अच्छा होगा। साथियों, अगस्त 2021 से जब हमने इस सीरीज़ को काउंसिल ऑफ साइंटिफिक एंड इंडस्ट्रियल रिसर्च इंडिया के इस कोलैबोरेशन में शुरू किया था, तब से लेकर के अब हम, जैसे मेरे साथी हरप्रीत ने बताया, 95th एडिशन पर हम पहुंच चुके हैं। इसमें हमने विभिन्न विषयों का समावेशन किया, जैसे कि शुरू हमने किया था हेल्थ से जुड़े हुए विषयों को, फिर उसके बाद हम न्यूक्लियर फ्यूज़न, प्लानिटरी मिशन, इंडियाज़ बैटरीज प्रोग्राम्स, सेल्स एनर्जी प्रोग्राम्स, जर्नी टू लंग्स, जर्नी टू हार्ट, सर्कुलेटरी सिस्टम्स, ऐसे विभिन्न विषयों पर हमने बात की। यदा कदा हम साइंस और टेक्नोलॉजी के क्षेत्र से भी बाहर निकले। हमने आर्ट ऑफ लिसनिंग पर बात की, हमने एंटरप्रेन्योरशिप पर बात की। हमारे साथ कई फेमस एंटरप्रेन्योर हमारे साथ इस प्रोग्राम में जुड़े थे। हमने नेवी ऑफिसर्स के साथ बात की। हमने अभी अगला शो भी, पिछला शो भी हमारा नेवी के साथ था, इस शो भी में हमारा नेवी के साथ, अगला शो भी हमारा नेवी के साथ होने वाला है। हम इसी तरह से चल रहे हैं। आपके हमें लगातार हमें मैसेजेस मिलते रहते हैं और आप हमें बताते हैं, आप हमें सुझाव देते हैं कि इन विषयों पर हम इस प्रोग्राम में समावेशन करें। आप अपने इन सारे प्रोग्राम, सारे सुझावों को हमें लगातार देते रहें। हमें बहुत मदद मिलती है और हमें बहुत उससे एक तरह से प्रेरणा मिलती है। दोस्तों, आज का जो प्रोग्राम है वो इंडिया साइंटिफिक कंट्रीब्यूशंस पर के ऊपर है, उसी क्रम में है। अगस्त 2021 में हमने एक प्रोग्राम, नवंबर 2021 में, आई बेग योर पार्डन प्लीज़, नवंबर 12, 2021 में हमने, जब हमारे साथ सहोत्रा सरकार ऑस्टिन से जुड़े थे, उन्होंने हरगोविंद खुराना पर बात की थी, ए लाइफ इन साइंस। आज फिर से हम एक बार खुराना साहब के ऊपर बात करना चाहते हैं। आज का प्रोग्राम भी आप लोगों के प्रेरणा पर है। आप लोगों के हमें लगातार मैसेज मिले थे कि खुराना साहब के साथ एक प्रोग्राम और किया जाए। तो हमारे साथ आज प्रोफेसर उमेश वार्ष्णेय हैं, बेंगलोर से हमारे साथ जुड़ रहे हैं। प्रोफेसर वार्ष्णेय, आपका बहुत बहुत स्वागत है। आपका हमारे इस प्रोग्राम में एनसीईआरटी के साथ कोलैबोरेशन करने में IISc का भी हम बहुत धन्यवाद ज्ञापित करते हैं और आपका भी बहुत बहुत स्वागत है और बहुत बहुत धन्यवाद। हमें पूरी उम्मीद है कि आगे आने वाले दिनों में भी भारतीय विज्ञान संस्थान से हमें इसी तरह से कोलैबोरेशन मिलता रहेगा। पूर्व में भी हमने कई प्रोग्राम IISc के साथ किए हैं और प्रोफेसर वार्ष्णेय बहुत जाने माने वैज्ञानिक हैं। अभी हाल ही में उनको राष्ट्रीय विज्ञान पुरस्कार के तहत पुरस्कार प्राप्त हुआ है राष्ट्रपति महोदया से, प्रोफेसर उमेश वार्ष्णेय, जिनकी मैं मुलाकात आपसे अभी कराता हूं दोस्तों। प्रोफेसर उमेश वार्ष्णेय obtained his bachelor's degree from the Jiwaji University in 1975. He obtained his master's degree in microbiology from the Govind Ballabh Pant University of Agriculture and Technology, Pantnagar in 1979. Professor Varshney obtained his Ph.D. degree from the University of Calgary, Canada in 1985. After his Ph.D. program, he continued at Calgary as an Alberta Heritage Foundation for Medical Research until 1988. Professor Varshney, he then joined at the Massachusetts Institute of Technology, Cambridge, United States for his second postdoctoral stint. Dr. Varshney joined the Department of Microbiology and Cell Biology at the Indian Institute of Science, Bengaluru in 1991, where he is still the Dr. A.S. Paintal Distinguished Scientist Chair and Honorary Professor presently. Professor Varshney is also an honorary faculty at Jawaharlal Nehru Centre for Advanced Scientific Research since 1999 and is a collaborator of the Genome, Metabolism and Biostruct Laboratory, Budapest. Dr. Varshney is known for his studies on protein synthesis and DNA repair in E. coli and Mycobacterium tuberculosis. These studies hold promise towards the development of tuberculosis vaccines. He has guided several scholars including 18 postdocs, 31 Ph.Ds and several master dissertations. He has around 200 research articles in various peer-reviewed journals and has delivered more than 150 research talks including several keynote addresses at many science conferences. Dr. Varshney is a J.C. Bose National Fellow of the Department of Science and Technology, Government of India, an elected Fellow of the Indian Academy of Sciences, Indian National Science Academy INSA, the National Academy of Sciences India and the World Academy of Sciences TRIST. He is also a recipient of the National Bioscience Award for Career Development of the Government of India. This was given to him in 1999 when the Department of Biotechnology was formed. The Council of Scientific and Industrial Research awarded him the prestigious Shanti Sarup Bhatnagar Prize for Science and Technology, which is considered one of the highest Indian science awards in 2001. And then in 2014, with the G.N. Ramachandran Gold Medal for Excellence in Biological Sciences and Technology for his contributions to biological sciences. As I said earlier, he was recently awarded the Vigyan Shri Award under Rashtriya Vigyan Puraskar scheme in 2024. So highly decorated scientist is with us. Professor Varshney, आपका बहुत बहुत स्वागत है। एक बार पुनः मैं आपका धन्यवाद ज्ञापित करता हूं और इंडियन इंस्टीट्यूट ऑफ साइंस का भी धन्यवाद ज्ञापित अपने NCERT की तरफ से और मैं अपने व्यक्तिगत तौर पर भी करता हूं कि आपका हमें साथ मिला। साथियों, आज के प्रोग्राम में जितने भी स्लाइड्स हैं, either they are belonging to Professor Varshney himself or they are taken from open sources, so those can be used for academic purposes as and when as any need arises. It will be nice that either you appreciate the collaboration and seek our permission for the purpose. So Professor Varshney, it is for you now. Thank you very much once again.
थैंक यू। पहले तो गगन जी आपका और हरप्रीत जी आपका बहुत बहुत धन्यवाद मुझे क्लास से जोड़ने के लिए जी। और गगन जी आपने तो बहुत ही जो है जेनरस इंट्रोडक्शन दे डाला और उसका आवश्यकता नहीं थी उसकी इतनी ज़्यादा, लेकिन धन्यवाद। तो आज मैं बात करना चाहूँगा खुराना और उनका जो रिसर्च था उसके बेसिस पे जेनेटिक कोड को कैसे सॉल्व किया गया। डॉक्टर खुराना का कंट्रीब्यूशन बहुत ही ज़्यादा रहा है, बिल्कुल अहम कंट्रीब्यूशन उनका रहा है इस जेनेटिक कोड को सॉल्व करने के लिए। और उसके साथ साथ में क्या बोलना चाहूँगा कि जो है प्रोफेसर खुराना का एक नज़रिया जो था वो बहुत ज़्यादा इम्पोर्टेंट है, हम सभी के लिए बहुत ज़्यादा इम्पोर्टेंट है। उससे बहुत सारी प्रेरणा हम लोगों को मिलती है और मुझे आशा है कि आज के प्रेजेंटेशन में आपको उसका एहसास होगा कुछ
जी।
तो पहले मैं आपको ये स्लाइड शो करना चाहूँगा। अभी हाल ही में 2-3 साल पहले उनका बर्थ सेंटेनरी ईयर था, तो उस पर काफ़ी ट्रिब्यूट्स उनको दिया गया है और मैं ये लेक्चर भी उसी श्रृंखला में उनको एज ए ट्रिब्यूट आई वुड लाइक टू गिव। काफ़ी लोगों ने देखा होगा कि 2018 में उनके बर्थडे पे एक गूगल ने एक निकाला था वेब पेज जो इंडिया में तो था ही, पूरे विश्व में था। तो उसको वो भी एक तरीके से उनको ट्रिब्यूट उन्होंने दिया और तो वो आप देख रहे हैं राइट हैंड साइड में। ऑन द लेफ्ट यू आर सीइंग अ पिक्चर ऑफ Hargovind Khorana and a long time associate of his Professor Uttam Rajbhandari. मेरा पोस्टडॉक का जो काम था वो मोस्टली प्रोफेसर राजभंडारी के साथ था, लेकिन डॉक्टर खुराना और डॉक्टर राजभंडारी के ग्रुप्स दोनों बिल्कुल एकदम साथ साथ काम करते थे। तो मुझे खुराना से भी काफ़ी मेंटरशिप मिली और उसके लिए मैं बहुत बहुत धन्यवाद करता हूँ उनका। और आप देखेंगे कि जो है काफ़ी कुछ उनसे सीखने को मिला और मुझे उम्मीद है कि आप सभी लोगों को highest type of efficiency is that which can utilize existing material to the best advantage. And this quote I like very much. In fact, when I started my laboratory in India, I put this quote on the shelf of the reagents so that people can make best use of what they were pulling out of that shelf. Now, this is the quote that I have been heavily impressed with throughout my life. And what I noted was that Khorana's life was actually also completely revolved around this quote. There's just a slight change in this quote here. Obviously, the highest type of efficiency is that which can utilize existing material or the available opportunity to the best advantage. And you'll see Govind Khorana as the best example of who utilized not only the material that was available to him, but also the opportunities that became available to him to the maximal advantage. So here is, in a nutshell, I think the earlier talk that was given by Sahotra Sarkar talked quite a lot about the life of Govind Khorana, which I am summarizing here only in one slide. And this you would have already seen in that earlier presentation, and if you have not seen, you will get to see this in many web sites. And basically, he was born in Raipur, which is in Pakistan now, and I think this town is now called as Ghazni. It's a small village actually, not even a town, which is called as Ghazni now. And he was born in 1922. Of course, I put a question mark here because no one really knows the actual date of birth. He may be older than what is written here actually. His father was a Patwari, and I think I don't need to explain this word Patwari here when I give a lecture in India. And he was very dedicated. He wanted to make sure that his family should be, his children should get good education. In fact, his family was the only family in the village of about 100 people which was literate. And so his father really paid a lot of emphasis on the education. Dr. Khorana's early education was at DAV school, Multan, which is, I think, about 150 kilometers away from Raipur. And in 1941 to 43, he completed his undergraduate degree from Punjab University, and 43 to 45, he did his master's again from Punjab University. And what I have actually shown here is that after he completed his master's, he actually got a fellowship. This is the period 1945. We were still under British rule. And during those days, this Commonwealth Fellowship had been started, and this was for higher education for people in UK. And Khorana was one of the recipients of the Commonwealth Fellowship. And at that time, they said that, this fellowship is given to you to study on insecticides and fungicides. But the High Commissioner in UK couldn't really find a PhD position for him to work on these two areas. But, he noted that Khorana had done his PhD in organic chemistry. And so he decided that, he could just do PhD in organic chemistry, and he managed to find one seat for him in University of Liverpool. And so that's where he did his PhD. From 1945 to 1948, he finished his PhD. And after completing his PhD in 1948, he decided to spend a year in Zurich, a very well-known institute, ETH. And he did that because he also wanted to learn some German, because, those days, a lot of literature, a lot of research in chemistry was actually published in German. And so he was really keen to learn German so that he could really get some more insights into the chemistry research. And so he spent a year at ETH, and this he did without getting a fellowship from ETH or the laboratory that he was working in. And after that, when his money ran out, he came back to India in 1949. But, during that time, this is soon after the independence, the job situation in India was not at all good. And he could not get a job in India. But he was lucky in the sense that he had developed some friendship with a person called George Kenner. And he was from University of Cambridge. And when he returned to University of Cambridge, he said that, there is a position available. And so I'll actually come to these things a little bit more. He joined back in UK as a postdoc. And while he was doing his postdoc, there is a scientist from Vancouver who was visiting here in Cambridge. And Khorana was, of course, looking for jobs. And this scientist, Shrum, he said that, look, I can give you a job, but I don't have any money for you to do research. But Khorana accepted the job, and Khorana then came to Vancouver. Khorana didn't even know where Vancouver was or Vancouver is, but he came and he started doing a lot of his research there, and he became really famous. From Vancouver, he moved to medicine in USA, and from medicine, he came to MIT. So this was his journey in very, very brief, and what you will now get to hear from me will be about what his teachings have been. And I'll come back to his research once again. But, this is important, and this has left a lot of impression on me, is that his own biography that he has written, A Life in Science, which was published in Science Journal. He says, I have met people who believed they knew at a very young age what they wanted to do in their lives. I envied them, but my own life was not like that. In fact, many of us are like this, that we do not know what we are going to do in future. And this is precisely what Khorana is saying, that doesn't really matter. Of course, Khorana also came from that kind of a background, in the sense that he did not know what he wants to, or what he would do in future. But you see, Khorana, what he achieved is something really amazing, that you will see. And so I think one should not really worry if you do not know what you're going to do in near future or long-term future. I think so long you take care of the opportunities that are available to you and you do your best, you will be successful. So this is, you will see now the opportunities and how he made use of those opportunities. And so while I bring here what he has done in terms of his science, I also bring it from the perspective of how he actually exploited and made the maximal use of the opportunities that were made available to him. And so in one of his own writings, he says that at the age 18, when I sought admission to Punjab University in Lahore, I applied to 2 departments, English Literature and honors course in chemistry. Now, Khorana was actually very shy. He did not want to face interviews, and admission in chemistry department required that he appears for interview, which he did not appear. But given his background and the kind of achievements that he already had, the chemistry department decided that they will offer him the admission anyways, and therefore there was a chance entry basically for him into chemistry. He would have, if this admission was not given to him in chemistry department, he would have just as well done literature and would have been doing something entirely different. But he got admission in chemistry department and he made maximal use of it. Maximal use of it in the sense that even when he was doing his masters, he actually published a couple of papers, which is very unheard of even today. But he achieved that. Now, the chance entry in chemistry sort of continues because after he completed his masters, and as I mentioned to you, that he got this fellowship, Commonwealth Fellowship, and again he was going to do something else, but the High Commissioner of India could find him admission in the regular chemistry department, and so he did organic chemistry then. Chemistry, organic chemistry, and then he completed his PhD in a remarkably 3 years' time. And as I mentioned to you earlier, he then wanted to spend a year at ETH to learn some German as well as to work with a very famous scientist there, Vladimir Prolog. And so that's what he did. And this again has a bit of a story here. And the story is as follows, that he knew that he wanted to work with him, but he did not write to him ahead of time. No one had ever written a recommendation letter for him. So he just decided to just go to Zurich, left his luggage at the railway station, and went to meet Prolog. And he said, okay, here is my PhD thesis. Please have a look at it. I will come the next day. And you can then tell me whether you can give me some space in your laboratory to work. And when he went the next day, Prolog said, okay, yes, welcome to the lab. And he was happy that, Prolog said that, yes, you can work in my lab. And this is quite interesting here. Khorana says in his own writings, looking back now over the years, I believe that spending a year at ETH with Professor Prolog is probably the wisest thing I ever did in my life. And this will, you will see why he says so. It is during this time that he was, when he was trying to look at the German literature, so he used to go and spend quite a bit of time in the library. And he used to sort of read the papers written in German, translate them to English. And during that time, he came across this, reagent, which is DCC. And this reagent actually became something that he would use maximally for all of his research. This reagent was known for quite some time actually, but it was sort of only published, and nobody actually sort of discovered it after it was published. But Khorana, because he was looking at the literature, he realized that this reagent can actually be made use of to sort of block the growth, make them active to do the reaction, and so he was really quite happy to sort of come across this molecule, and he tried to make use of it even in the lab of Pralog, and he was partially successful doing that. But then, as I said, he would use it at a later stage. So then this is the story that I've already told you, that after 1 year, spending 1 year in ETH, he returned to India. And at that time, of course, there were no jobs for him. There were no jobs for anybody, actually, not only for him. And this was really quite sort of disappointing in a way. But I think sort of worked out quite good at the later stage. And what you will notice here is that he ended up in Cambridge. And he says that the Cambridge was really exciting place. It was an exciting place because one of the very first protein was to be sequenced by Frederick Sanger, and of course he got the Nobel Prize for it. And Max Peruz and John Kendrew, they were solving the structure of globin and hemoglobin, and these are all textbook material. And when you read anything about globin, hemoglobin, you'll come across these two names and the structure of hemoglobin. And Watson Crick, they were trying to solve the structure of DNA, and so Khorana landed in Cambridge after not finding a job in India in 1949. He returned to Cambridge, and he went into an environment which was really very, very inspiring. And I think a lot of influence of biology, I would suspect that he was influenced by this research that was going on in Cambridge in his future career there. And so he says that the molecular biology was in making. And this is really, I think, very, very interesting, because he realized that the molecular biology was going to be something which was to be something going to be in thing, and he would work on this in this field. So as I mentioned to you, during this time when he was working in Cambridge, Dr. Shrum, who came from Vancouver, he was looking for people. In fact, a British Council they had established, but they were not finding good scientists. And one of the reasons was they didn't have a whole lot of facilities and money in research grants. So Shrum, when he took it as a director of that place, he decided that he will get some money, but then he wanted to hire really, really bright people who could actually do some research there. And Khorana came into his picture. He offered the job to Khorana, and this is where actually in British Columbia, Vancouver, he started working on nucleotide synthesis. This was again a really sort of in topic at that time, because nobody had synthesized these nucleotides, for example, ATP. And the reason was that, it was very difficult because there are multiple reactive groups. And so if you do the chemical synthesis, you really need to be very careful in protecting the groups that should not react at a given time. And so this is where I think Khorana's expertise came into picture, and the reagent that he had suddenly discovered in Switzerland. And because he was working with these nucleotides, he also came in good contact with Arthur Kornberg, the DNA polymerase person, and Paul Berg, that most of you would actually know because of the recombinant DNA technology. Both of these actually became his friends. They visited his lab, and then he also visited their lab, and so the friendship really started. And in this picture, you'll see Arthur Kornberg and Paul Berg here. at a party that Khorana is there. Yeah. So he developed a really good friendship with them. And that friendship turned out to be very helpful to him, because when he was trying to synthesize the polymers of DNA, he made use of the reagents that these people had developed. This is actually a picture of Khorana's group. Khorana also had this sort of legacy that every year that he would have a picture of his group. And this is a picture that was taken in 1959. At that time, he was in Vancouver. And the reason I show you this picture is, of course, you see Khorana here, but next to him, you see Mike Smith. And Mike Smith actually worked on site-directed mutagenesis, and he also got a Nobel Prize. So that's one of the reasons I thought I will show you this picture. Khorana sort of, had the knack of realizing, what really is going to be exciting. And you'll notice that Khorana worked with Vladimir Perlog in Zurich. He got a Nobel Prize in 1975 for stereochemistry of organic molecules and the reactions. He worked with Alexander Todd in Cambridge, and he got a Nobel Prize in 1957 for the synthesis of nucleotides. And his postdoc, Michael Smith, got a Nobel Prize in 1993 for site-directed mutagenesis. So he always was in a company of really, really knowledgeable people. In Vancouver, they even actually established a park in his name, Khorana Park. And he was even happier to note that there was another park right next to it, which was established by the name of Michael Smith. So this was really something that he enjoyed. After he completed his work of nucleotide synthesis in establishing friendship with a lot of people, he decided to move to medicine. At that time, University of Wisconsin made him a fantastic offer, which he could not turn down, and he moved his laboratory to Medicine, USA. And this is where quite a bit of his work, actually I would say most of his work relating to genetic code was done. And so this is really, again, something that he did here. You can easily call it a synthetic biology. So the roots of synthetic biology are really in what he did at medicine in University of Wisconsin. And of course, you would all know that he got the Nobel Prize in 1968 in the field of medicine of physiology. So now let me just tell you a little bit about how this genetic code was actually solved. And I show you this picture again. Here you will see Khorana, and this is Craig, and you see this lady here. This is Marian Grunberg Managua, and I chose this picture because I think all of these guys have a lot of contribution in solving the genetic code. This is a picture from Cold Spring Harbor, and let me just tell you what the sort of basis for genetic code is. Now, it was clear that the proteins largely have 20 amino acids, and DNA has 4 nucleotides. And so at that time, usually because with Crick and Gamow, these guys used to discuss a lot of things. George Gamow is actually a mathematician, physics person who sort of ventured into this genetic code story for a little while. And what he reasoned was that if the genetic code, that is how the nucleic acid code or make the protein, how is the language of nucleic acids translated into the language of amino acids. If there are only 4 nucleotides, how are they coding for 20 different amino acids? So if there was a single nucleotide which was being translated into a single amino acid, the maximum number of amino acids that you could code for would be only 4. But if there were 2 nucleotides that were being read at a time, then it could be 4 times to the power 2 will turn into 16 different combinations, which was still not enough to code for 20 amino acids. And so then he thought of the 3-letter code, and that could actually give 64 unique combinations of the nucleotide sequences, and that could actually suffice to code for 20 different amino acids. And that was actually, this theoretical calculation was in fact correct. Now, this was, all of it was theoretical calculation that a triplet nucleotide would be coding for an amino acid. But there were biological evidence also for it, and which was again sort of gathered by Craig and Sidney Brenner. I won't go into the details of it, but to sort of sum it up, what they did was that if it is a triplet code, and if there was single nucleotide deleted, the gene will become non-functional. If the second one was deleted, still the gene will remain non-functional. But if the third one was deleted, then the reading frame will be established once again, and therefore the gene will become functional. And it sort of worked for them when they were working with the mutagenesis of FAST-T4. And so that was sort of, biological or the genetic evidence to say that the genetic code is actually a triplet. So that was the background that, people were looking for a triplet genetic code. But the real story actually sort of starts with this work of Marion Grunberg Managua that you see over here, a French lady. She was working with Servo Ochoa, who was also a Nobel laureate. And as the serendipity will have it, they actually were working with an enzyme called polynucleotide phosphorylase. Now, this enzyme is actually an enzyme which degrades RNA. But it turns out that if they would have high concentration of the dinucleotides, as is shown here, PPN and PPN, it would actually polymerize them, will make a dinucleotide and release a free phosphate. Normally, the reaction will go into this direction, but because the concentration of these products anyway is high here and not having free phosphate, the reaction goes into synthetic mode. So this was actually a very, very important discovery, and that sort of led them to synthesize an RNA molecule in a template independent manner, as you would have studied and would know that RNA is actually made utilizing a template of DNA. Of course, there are other mechanisms as well, but mostly the DNA will be used as a template, and you'll get the RNA. But here it is template independent synthesis of RNA, and this importance of this finding was realized. And the very first thing that was done by Marshall Nierenberg and Mathai, Nierenberg is another person who got Nobel Prize along with Govind Khorana. They actually synthesized a polymer of uracil. So you have a poly-U synthesized, and at that time, protein synthesis machinery was already known. People already knew how to translate an mRNA into a protein. And so when they introduced this RNA molecule having poly-U into translating system, they got a peptide which had only phenylalanine. So the very first genetic code that could be solved was that UUU codes for phenylalanine. So this could be done quite readily for poly A, could be done for poly C, could be done for poly G, but one would get stuck at this stage that you wouldn't be able to solve the genetic code beyond solving the 4 codons. So at this point, there was another kind of possibility that they started looking at it, which was just going by probability. So if we take so in the very first slide, where I was talking about Marion Grunberg Managua's work, if you took 2 nucleotides, it will polymerize it. Now, what these people did, okay, let's try taking 3 times uracil and 1 time guanine. And so you could make a polymer which would be either UUU, UUG, UGU, GUU. All of these combinations would be possible, and you could calculate the probability. So the probability of making a triplet U would be 3 by 4 times 3 by 4 times 3 by 4. So this will be 27 bar 64. And if you set this as 1, you can calculate the relative probability of all of these combinations. Now, this was actually good in a theoretical sense, that you could do this, that you could make a sort of genetic code which consisted of G and U, and you could even calculate the probabilities of these different codons, because see, if you have less G and you try to look for the codon which is GGG, it will be the least probable combination that one would get. But nonetheless, these polymers could be used, and then one could find out what are the amino acids that are incorporated because of this polymer, and you can get this ratio. So many of these combinations, you see the probability matches. Perhaps these 3 code for these 3 amino acid, and this is coded for by this, that GGG codes for glycine. Now, this in the retrospect, of course, it is correct that GGG codes for glycine, but to determine, if you do not know what codon codes for what amino acid, it will be next to impossible to sort of assign that GU codes for a particular amino acid there, that GU, for example, codes for valine. So this sort of got stuck at this position. You could change the ratios of the nucleotides that you're using for this polymerization, but nonetheless, every time you will get into this mess that you will have a series of amino acids that are incorporated by the polymer, and you wouldn't know which codon is coding for which amino acid. So at this point, Khorana's entry came into picture, because as I mentioned to you, Khorana had already developed methods to synthesize nucleic acid with the known sequence, that he knew that this is the first nucleotide. If you push the next nucleotide, that will be the second and third, so really, I think that Khorana was always thinking ahead of his time. And Khorana actually wanted to work at the interface of chemistry and biology right from the beginning. And so he actually decided that he was going to take try this problem, try to settle this problem by synthesizing sequences of the nucleotides with known sequence. And so this is what he did, and I think this maybe I will just briefly mention it, but you can read this in his Nobel lecture. So what he did was, he was able to synthesize short nucleotide, oligonucleotides. I mean, he could do longer ones also, but this synthesis takes a lot of time. And so he used short nucleotides, and then he made use of the Kornberg's enzyme, which is DNA polymerase, to convert them into longer polynucleotides. He could then make use of RNA polymerase to make the RNA out of this DNA, and use this RNA to make protein. He knew the sequence of this RNA, he knew the sequence of this polypeptide, and he could correlate which sequence corresponded to which amino acid. So this was really a really nice thought from his side. And he used a lot of tricks also, actually. So let's say he synthesized a poly T. This will pair with poly A. And you see, there are 11 T's and 7 A's. So there will be a gap generated. That gap can be filled up by polymerase. So he can extend this A7 into A11. Now, if he melts it again and re-anneals it, again there will be sort of gaps in the ends, and he could fill them up again with polymerase to make a polymer out of something which was small. Now, to make a polymer, he used the nucleotides which would pair. So let's say T will pair with A and A will pair with T. So he will get poly DA-DT. Likewise, he can make polymers of TGAC, he get poly TCCA. And so he used trinucleotides polymers, and then he could make polymers of 3 different three nucleotide sequences, repeating sequences, and so on. So this was his initial thing to generate the polymer. And now you see, he still has a mix here. So because DA and DT. Here Tg and Ca. See what he did next. Now if he uses this as a template and in the reaction only uses ATP and CTP, he will make only Ca, because the complementary template Tg will serve as a template and Ca will be made in large amount. Likewise, if he used only UTP and GTP, he could make poly UG. Now, if poly Ca he uses in translation system, he will know which amino acids are made. Poly UG, he will again know which amino acids are incorporated into the polypeptide. So, like that, he actually used the tricks of translating only or transcribing only one of the strands of the DNA and the second strand in the second reaction. So, he was able to, here is say, double stranded DNA, but he was able to then convert it quite readily into single stranded RNA of the known sequence. Likewise, he made many more synthetic ribopolynucleotides of repeating nature. So I talked about, dinucleotide repeats, trinucleotide repeats, and tetranucleotide repeats that he made. Using these, he did some, again, beautiful experiments, really very simple experiments, and I think this is something that we should all aim for. Simple experiments, fantastic interpretations, you can't go wrong. So here, what he's doing is using the UC polymer for translation. And you see that, there is a radioactive leucine here, and he uses serine, which is in the reaction. If he has both leucine and serine, you see the polypeptide is being made like this. If he leaves out the polymer here, then the synthesis is weak. If he leaves out here, and if he leaves out one of the amino acids, then again the synthesis is low. Now he can do the other experiment where he can take the serine radioactive, but leucine is cold in the reaction, and he again sees the synthesis of the peptide and the controls where he leaves out either the polymer or the amino acid, he doesn't get synthesis. Now, what he did was, I've given you the example with 2 amino acids, but this kind of reaction he would do with all the amino acids. And what is shown here is the one that has worked. So you will notice that, I highlighted this one. So let me just read out what he has said. Incorporation of only 2 amino acids is observed, which means that this polymer codes for only 2 codons of the 2 amino acids here. Incorporation of one of these amino acids is dependent on the presence of the second amino acid. When he leaves out serine, the leucine, he doesn't get synthesis here, and if he leaves out leucine here, he doesn't get synthesis. So if he doesn't take both the amino acids, synthesis of the full length polypeptide does not take place. And the incorporation of the 2 amino acids equimolar. He can calculate the molarity based on the amount of radioactivity incorporated, and he can see that they are equimolar in amount. Now, if you try to put all of these observations that he has made from this experiment together, the only thing that you will come up with will be that there are 2 codons, UCU, CUC, UCU, CUC, UCU, CUC. Because the incorporation of this is dependent on this, they are equimolar, this. So you can fulfill all of these conditions if you now assume that the polypeptide is being synthesized with alternating amino acids, and alternating codons will be there, and that is the way that you will get it. So like that, he made use of these polymers. He found out what amino acids were incorporated, and that allowed him to sort of figure out which codons code for which amino acids. Now, here again, it's not a perfect answer, because with this experiment alone, he would not be able to tell whether UCU codes for serine or leucine. But then in future, he would be able to design more experiments and then solve that UCE is this and CUC is this. So this way, he was able to solve the genetic code, and it could be done simply because he could synthesize the polymers of the known sequence. This was the key in solving the genetic code. Now, the other thing that really happened, And this is the work which was actually set up in the lab of Nirenberg, but he also used it. And what Nirenberg found was that if he takes a triplet sequence of the known nucleotide sequence here, which again Khorana can only make, he can actually trap the tRNA. So let's say you have AUG, you will trap a tRNA which has CAU as anticodon, and he will be able to find out what amino acid is carried by this tRNA. So in doing so, you could actually make use of the triplet nucleotides and find out which amino acid of which amino acyl tRNAs were captured on the ribosome. And this also allowed them to find out which triplet coded for which amino acid. Now, there is a problem with this experiment because the background, journal background is quite high, but I think combined with his earlier work and knowledge from this work then allowed them to solve this entire genetic code, which we routinely see in our books and wherever the genetic code table, where now we know that UUC code for, are coded for, the code for phenylalanine, leucine, methionine, and so on. So this sort of, was celebrated, and of course, this is what was responsible for the 1968 Nobel Prize. Now, normally one would think that, if there are 64 codons, and of course there are some stop codons. In fact, let me just go back a little bit. See, when he used the tetramers, for homotetramers here, he didn't get polypeptides. He got some amino acids incorporated, but polypeptides he did not get. And indicated to him that there are sequences in the genetic code which indicate termination codon. So the concept of termination codon actually came from this experiment. And so he was able to then decipher the stop codons. So there are 61 codons that code for amino acid in a sort of a standard table. And these 61 codons are read by tRNAs of 32 different kinds in journal. In Neurospora, there are only 24 that read the 61 codons, and mitochondria, there are only 22 which read the 61 codons. So this genetic code table that you're all familiar with was established. Now, there was a lot of discussion that, the genetic code once evolved is now frozen because, if you try to evolve the genetic code, the organism will have to pass through death, and that is not possible. So there was a hypothesis that it's a frozen accident, but you see that is not so. There are a lot of evidence now that the genetic code is still evolving, and the meaning of these codons is not always universal. The mitochondria and in fungi, for example, some of the codons are coded differently. So, for example, CUG is actually leucine. CUG in Candida species codes for serine. And so there's already people knew that there are some altered definition of the codons that the organisms utilize. And with that, there are also combinations that many of the termination codons actually code for amino acid. Selenocysteine, which is one of the sort of rare amino acids that is present in the proteins, is coded for by UGA. There's another amino acid, pyrrolysine, that is coded by UAG codon. So there are many organisms which utilize the termination codons for decoding them as amino acids, and likewise there are some codons which are decoded as termination codons. So there is actually the genetic code is actually still evolving. Now, one of the huge achievements that Khorana did, and I mentioned to you that most of the time they were synthesizing short oligos because synthesis of the oligos took a lot of time, and so synthesis of large genes was a very, very tough time. It would take years and years and years of a whole battery of postdoctoral fellows. So one of the things that Khorana did was he was interested in synthesis of total synthesis of a gene. The very first gene that was synthesized was tRNA, that codes for alanine. Now, this story did not move too far simply because they could not really express it in the cell. But then, the total synthesis in all these, genetic code itself sort of, earned him the Nobel Prize. And here are the Niren Hawley, who shared the Nobel Prize with him.
Wrap it up in next 2 minutes, sir. Please.
Will be done. Yeah. So he got a lot of these awards, and you will notice that the government of India gave him the Padma Bhushan Vibhushan Award, and he then moved to MIT. He started working on again difficult problems of membrane proteins, which bacteriorhodopsin and rhodopsin. He then, at this time, he decided to synthesize, chemically synthesize the gene and show that this will work in biological system. And this was actually the gene that tyrosine tRNA, which could actually function in E. coli, and he synthesized that. There's a lot of story about this, but time is limiting, so I won't go into it.
Yes.
Started working on the membrane proteins. And as I said, that he used to take these pictures. I was there in 1988 to 1991. This is a picture in 1990. You'll see me over there. I was visiting in 97, another picture. And Khorana was not only just work. Actually, he used to call his people to his home for parties. And you see, he's standing on the road showing which way to go.
Right.
He's enjoying the party. Again, he's enjoying party. He used to also go for fishing. You'll see that I'm also there. I even caught a fish there, deep sea fishing. This is a symposium that was held in his honor in Waltham, Massachusetts. He actually put together a book on his work, and he actually gifted it to many of his colleagues. And you see another symposium that was held in his honor in 2004 in Japan, which I also happened to attend. There was another symposium held 2009. He used to enjoy these things, the talks that were given at these symposia. So the symposia held in medicine 2009, and then 2011, the year that he passed away, November 9, we had a symposium for Raj Bhandari, but because he had passed away just then, we had a special session for Khorana at this meeting. There was an issue of resonance, which many of you will be familiar, which was brought out to highlight the work of Khorana. Then this is probably the last symposium that was held in the honor of Khorana, which was in Chandigarh, Mohali, and in 2017. Khorana is extremely well known for his, the fellowships have been started in his honor. And I think I'll spend a little bit of time here. Please give me 1 minute for this, because I think this is very important. There are a lot of things that you can learn from Khorana. Basically, he made use of the opportunities that were given to him. But, there are things, he always said that be determined and self-motivated. There may not be people who would try to motivate, but if you are determined and motivated, you'll achieve what you want to. Be bold and aim high. And you will see that he always aimed high. He thought of futuristic problems and solved them. Be disciplined and work hard. Always do your best. Strive for excellence. This is really very important. Stay focused. Do not get distracted. Plan carefully. Prepare yourself well. Be thorough. Follow every lead. No shortcuts. This is very important. Again, the researchers these days, they want to make shortcuts. But shortcuts may not always work, and they actually may lead you in the wrong direction. Do everything necessary to prove a point, and persevere. Don't give up. That's very important. And many times we are doing something very, very novel. People may not appreciate your work, and you may have to do it all alone, in spite of the fact that people will say that, look, you're not going to be successful. So this is what Khorana was.
Thank you, sir.
Thank you.
Thank you, sir. Thank you, sir. There are many takeaways from today's sessions. Today's session, yes, sir. We are really running short of time. We have got several questions, but I will just take a simple quote from Kumari Usha, one of the viewer. She says that the session is just something like listening a Ramayana story. However, I personally believe that it is not only that, it's much more than that, listening a Ramayana story. It's much more than that. I really had many questions. I will send you those questions, sir, and to seek answer, and we'll try to send it to the person, those who are asking the questions.
Okay.
Thank you very much once again, Dr. Varshney. We really enjoyed listening to you, particularly about Khorana's coding, synthetic biology. We received many questions about the synthetic biology and then its relationship in today's life. And I will get back to you for those. Sir,
Sure.
So this was today's session for us, today's friends. Harpreet, please take on.
Of course, first of all, thank you so much, Dr. Varshney, for your immense contribution to the session. The language was simple, even I could understand the concepts. Thank you for the same. And now, let's just get to know about what's coming next. Next week, we have a special session, sir. So why don't you tell us more about it?
Yes, cool. Friends, next week on 31st of January, same time at 10 o'clock, we will be having CDR Commodore Kalish Mohanan with us, and he will be talking about the Royal Indian Navy and its role during the transition in India's independence. So this is again going to be a very thrilling session, very exciting session. Be with us at the same time. Looking forward meeting you again on 31st of January 2024, same time 10 o'clock in the morning.
Thank you so much, sir. We are looking forward to it, and also we are looking forward to your comments and suggestions. And that was our 95th edition of special webinar series, Listening to Learn, and we talked about Khorana and the genetic code. And thank you so much, Dr. Varshney, once again. Thank you so much, Dr. Gagan Gupta, for your contribution to the session. It was indeed pleasure. And now, before I wrap up, let's take a very important announcement out here. It's regarding the All India Children's Educational E-Content Competition. Well, 31st of January is the last date. It has been extended, so you can always send in your entries to us. You can log on to CIET website, which is www.ciet.ncert.gov.in for more details, or you can scan the QR code on the screen. And now let's take a quick look at the schedule, what's all coming ahead. Well, we started the day with Yog Shakti, and we are through with our special webinar series, Listening to Learn. In a short while from now, you can watch our session on teaching learning interventions at 12. 2 p.m. is the time for session on mathematics, followed by a session of social sciences at 2:30. 3 p.m. we will bring a session in Sanskrit, followed by a session in sciences, and simultaneously you can watch our session Manodarpan Paricharcha also. 4 to 5 p.m. is the time for our online training program, and from 5 to 5:30 we will bring you Sahyog, and at 5:30 we will bring you a session on school leadership development. With this, it's a wrap. This is me, Harpreet Kaur, taking leave of you, and join you soon. Namaskar.